Hello, everyone. So we will going to discuss about Enterococcus Faecalis. I'm one of the reporter and I am Liniel M. Resus.
And I am the second reporter. I am Alea Zarina Disiton.
And I am Sid Polindo Rosal, and I will be the last reporter.
Enterococcus Faecalis. So first is I'm going to discuss about the general characteristics and the epidemiology and pathogenesis. So first is the general characteristics. Did you know that E. faecalis was also known as Streptococcus faecalis? Scientists previously categorized the bacteria as part of genus Streptococcus. In microscopic appearance, E. faecalis is ovoid diplococci and cocci in clusters. In liquid media, they grow in chains or formerly classified as streptococci. Optimal temperature for growth of E. faecalis is 35 degrees Celsius. E. faecalis is a normal inhabitant of the intestinal tract, female genital tract, and less commonly oral cavity. It is a gram-positive commensal bacterium inhabiting the gastrointestinal tracts of human. Like other species in the genus Enterococcus, E. faecalis is found in healthy humans and can be used as probiotic. Number two is the epidemiology. Large number of studies showed that E. faecalis can be recovered from beach sands, soil, aquatic and terrestrial vegetation, fresh water, and marine water sediment. E. faecalis infections spread from person to person through poor hygiene. Because these bacteria are found in feces, people can transmit the infection if they don't wash their hands after using the bathroom. The bacteria can get into food or into surfaces such as doorknobs, telephones, and computer keyboards. Pathogenesis and spectrum of disease. E. faecalis is found in most healthy individuals but can cause endocarditis and sepsis, urinary tract infections or UTIs, meningitis, and other infections in humans. Enterococcus faecalis cause a variety of infections: prostatitis, intra-abdominal infection, cellulitis, and wound infection, as well as concurrent bacteremia.
I will be now discussing the laboratory diagnosis of Enterococcus faecalis. Specimen collection and transport. The specimen used for Enterococcus faecalis is mainly feces and rectal swab. People can transmit the infection if they don't wash their hands. In specimen processing, the collected feces and rectal swab specimens are surveilled during a period of 8 months. Direct detection and methods. Isolates like Enterococcus faecalis that are highly resistant to vancomycin can be directly detected as vancomycin resistant when tested by conventional antimicrobial susceptibility test methods. A change in color of the medium surrounding a colony from pale yellow to black indicated esculin hydrolysis, and esculin hydrolysis signifies a colony of Enterococcus faecalis in the supplemented via Agar. Pyrrolidinol peptidase test, a gram stain which shows gram-positive cocci on the plate, and a gram-positive identification GPI card were used to directly identify Enterococcus faecalis. For the approach to identification, the test suitable for the identification of the Enterococcus faecalis is the Vancomycin Bhi screening or the Vancomycin Agar screen plate that contains brain-heart infusion agar that is supplemented with 6 microgram per ml vancomycin, which is very useful in screening for vancomycin resistance. The bacteria is known to be susceptible to vancomycin and to high levels of gentamicin and streptomycin. Synergy screen test as negative. that made this test very suitable for the identification of Enterococcus faecalis. This screening test can also be used as synergy screening for Enterococci and S. aureus. For its cultivation, Telluride blood agar is a selective differential enrichment agar useful in isolating Enterococcus faecalis alone. Tubes and base medium can be stored and melted to make the complete medium as culture plates as needed. Freshly plated medium should be inoculated, streaked for isolation, and incubated at 35 degrees Celsius. In serodiagnosis, the E. faecalis specific antigens of 40 and 37 kDa, which are prominent in endocarditis, were strongly expressed following growth in serum or brain-heart infusion, but not after growth in a chemically defined medium. The expression of these antigens in vivo was demonstrated in cell grown as a biofilm on celastic disc using the peritoneum of rabbits.
And for the fourth part, we will be talking about antimicrobial susceptibility testing. Enterococcus faecalis has both natural and acquired immunity from antibiotic treatment. It can survive in a range of temperature and acidic or alkaline environments like the stomach of animals and humans because Enterococcus faecalis is an opportunistic bacteria that is a part of the normal microbiota of the human gut. So Enterococcus faecalis can resist many antibiotic treatments because of their biofilm formation, nutritional needs, penicillin binding proteins, and environmental folic acid absorption, which we'll be discussing on the next slide. So biofilm formation. Enterococcus faecalis can bind together to form a thin but sturdy layer. This is known as the biofilm. So this biofilm helps the bacteria to stick firmly on the surface. Then next, we have the nutritional needs. Enterococcus faecalis can endure long periods of time without nutritional source. It can even use the serum in the blood as a source of nutrition. Then we have the penicillin binding proteins. So these proteins make Enterococcus faecalis naturally resistant to penicillin. So instead of killing the Enterococcus faecalis, penicillin only inhibits the growth of Enterococcus faecalis, but does not completely destroys the bacteria. Then lastly is the environmental folic acid absorption. Bacteria requires folic acid to grow. Most cannot absorb it from the environment, so most of the bacteria make their own. Medications that interfere with the production of folic acid can often destroy bacterial infections. However, Enterococcus faecalis can absorb folic acid from the environment, so this medication have little effect on the bacteria. Then lastly, we'll be talking about the prevention. Enterococci are mostly transmitted due to poor hygiene. So this includes Enterococcus faecalis. It is naturally present in the gastrointestinal tract and is found in the fecal matter. Improper cleaning of items containing fecal matter or not washing the hands after restroom use can increase the risk of bacterial transmission. So here are the preventions. One, we must sanitize our hands and avoid sharing soap bars. Everyday objects such as telephones, doorknobs, towels, and sharing of soaps also contain Enterococcus faecalis. Then we should also sterilize medical tools and things that are susceptible to the bacteria. Because if not thoroughly cleaned, catheters or dialysis ports can also transmit this infection. And lastly, the bacteria can also spread throughout the hospital if healthcare workers do not wash their hands. So not only the healthcare workers, but including us, we should wash our hands to prevent Enterococcus faecalis. So here are the references of the report. We have this, and that. So that's all. And thank you for listening. God bless.
Tas ikat na ba kay makakat baka video?
So, ano man guys, okay na to?
Nasi